All right. Thank you to the organizers for giving me this opportunity to present my work. I'm Brian Linge. I will be presenting meta-analysis of gene expression, disease signatures, and colonic biopsy tissue from patients with ulcerative colitis. So this is my disclosure. And so for those of you not familiar with Elementive, Elementive formerly was known as Robarts Clinical Trials and underwent a name change. We are a niche gastrointestinal contract research organization that provides a wide range of clinical research services for both academic and industry clients, and we've been around since 1986. So in addition to providing those CRO services, we also reinvest our profits into a research component of the company. So this research component works to help improve outcome development, starts and works with investigator-initiated studies. There's also a precision medicine component, which will be talking about today as part of our team's work. And finally, all of what the research group does is published in peer-reviewed journals and is available for the public free of charge. So part of this precision medicine team is the Elementive Translational Research Consortium. So this is a global group of scientific experts in the IBD field who work with translational studies. And through this work, I'll be presenting the work today, and they are, we'll mention them by name at the end of the presentation. So as a team that works with pharmaceutical companies and biotech companies interested in target identification and biomarkers, one of the main questions that you could imagine would come up would be a very general one, which is my biomarker differentially expressed in ulcerative colitis compared to healthy controls? So an extension to this you could imagine would be, is my biomarker responsive to treatment, or is my biomarker predictive of whether my patients will respond to my drug or not? And so in all these cases, depending on the type of marker you're looking at, there may be many different data sets which you can look to answer this question. Now, because there's many different data sets, hopefully they would all agree and they would give you the same answer. And unfortunately, that's not usually the case. So if you're lucky, you may see yes always or no never. It's not regulated. But in most cases, the answer is more nuanced to answer, like sometimes. And this is because not only is there differences in technology and the ability to precisely measure these biomarkers, there's always also differences in disease severity or disease attributes that may contribute to the answer. So the question we were interested in looking at is, how do we answer such a question by combining the data from individual data sets? And how do we come up with an answer that is satisfactory from a conciseness point of view, but also from understanding the variability in that answer? And so this is a task that's suited for meta-analysis. So specifically, when we're doing meta-analysis for differentially expressed genes or differential expression of a certain biomarker, how do we look to combine those many experiments? How do we combine them? And so there's a multitude of meta-analysis methods that one can use. And for situations such as differentially expressed genes, the most common one is most likely to be taking an intersect or a union of the lists, such as a Venn diagram. So in that case, you would just look at genes that are differentially expressed in each data set, and then look at the overlap between them. Alternatively, you could look at the union or the combined list and say those are the genes that are differentially expressed. Other methods include taking a vote or consensus. You could directly combine all the data sets into one superset and then treat that as an experimental set. You could combine ranks, and you could also combine p-values. Now, all of these have their different strengths and weaknesses, but the sixth one that we are using here for this analysis is combining effect sizes. And specifically, we'll be using a random effects model to do this. And while I'm not going to get into any of the statistics involved in this analysis for this talk, what I will say is one of the great benefits that we thought this type of analysis provides is shown here on the right, where an example of a type of study uses a random effects model, where you would have on the y-axis different studies, each with their own mean as well as 95% confidence interval, which is shown by the dot and the whiskers. So in this, you can see the behavior of the endpoint of interest in each one of the different lines as you go down. And then on the bottom, you can see the effect of the random effects model, showing that you have a mean that is somewhere in between the combined results of the individual data sets. And as I'll mention more later and show you some examples, we find this is a very useful way to combine data sets and understand not only what the true value may be, but also understand the variation between these values. So if we get back to this question, is my biomarker differentially expressed in ulcerative colitis compared to healthy controls? We wanted to take a look at this somewhat basic question and see how meta-analysis can help us give an answer to this, that we are satisfied from not only a conciseness point of view, but also retaining the information from the individual data sets. And so when we looked in the public domain to find what data sets were available, we found 85 different data sets that filled the initial search criteria. So we looked on the NCBI Gene Expression Omnibus, which contains hundreds of thousands of gene expression data sets, and you can filter those by different qualifications. For example, we were interested in looking at ulcerative colitis samples from humans, and then we were interested in this analysis only to look at microarray. And we did this by design to decrease the variability we might expect from different technologies such as RNA-seq. Then we went through a series of filters to make sure that the samples were comparable. So we made sure that there were no isolated tissue types in the first, and that removed 17 data sets. We removed very small data sets which had less than 10 total samples. We made sure that they had a healthy control, which we could use to compare within that data set. And this is an important point, there were several data sets that had samples used in multiple versions of that data set. And while this on its own is useful if a investigator wants to update their previous data sets, for the purpose of the meta-analysis, it was important to remove this to make sure that there was no redundant data sets. Finally, we made sure we looked at adult samples, in the pediatric samples were removed. We only used samples from inflamed biopsies. And lastly, we made sure that the platform normalization was compatible with what the downstream processing we were interested in. And so that gave us a final of eight data sets. So here I showed those data sets. On the y-axis, I'm showing the gene expression, the geo series accession number with the sample source, the publication year associated with it, the number of samples, both for the UC and the non-IBD control, biopsy location, sample storage method, array platform, disease duration, disease activity measure, and then C-reactive protein, disease extent, and medications. And so as you can see here, there's a wide range of disease number of disease samples between 8 and 87, and a wide number of controls between 5 and 21. Additionally, there's various disease metrics that were used in each one of these studies. Because there was no standardized way to report this data, sometimes endoscopy values were reported, sometimes Mayo Clinic scores were reported, and sometimes neither of those were reported. There was a range of sources of these. One was from industry, 5 were from Europe, and the rest were from the United States. And lastly, I'll point out that while we were able to determine some of the patient demographics from the population data, very few of these data sets actually had individual patient level demographic data and disease activity metrics. So with this limited us in the ability to look and subset those data sets, which I'll talk a little bit more about later. So what we did is take these data sets and put them through a single analysis pipeline to see how many differentially expressed genes were in each one of those data sets, and then perform the random effects model to see what the meta-analysis tells us about those numbers of differentially expressed genes. And so that's what I show in this slide. I'm showing the number of differentially expressed genes On the y-axis, the ones in black are upregulated in the UC compared to non-IBD control, and the ones in gray are downregulated. And this is using the same cutoff of adjusted p-value of less than 0.05 and an absolute log fold change of greater than 1.5. So as you can see, there's a wide range of number of differentially expressed genes across these data sets. And when we look at the meta-analysis of those 8 data sets, we see that you get an overall 1.2 times lower and 1.7 times lower downregulated genes compared to the individual data set median. So you're basically getting a reduction compared to some of the data sets and an increase compared to others. And I'll show you a little bit more in detail how this looks from the individual gene point of view. So this is a volcano plot showing the log 2-fold change for individual genes that pass the meta-analysis cutoff, as well as the negative log 10 p-value, which is inversely related to the significance. So the dots in red are significant from a log 2-fold change point of view, as well as the p-value point of view. And the ones in blue or green do not match one or both of those cutoffs. So the point of showing this is just to indicate that the levels of some of these genes, such as reg1a, are very highly regulated, whereas genes like claudinate are very downregulated on the left. So to get an understanding not only at the individual gene level, but how these individual data sets compare to each other, we perform principal component analysis. So this is used to understand the variability between the different data sets. And so the variation in the experiments is visualized on principal component 1 and 2 in the X and Y axis. And as you can see here, there's a large spread in the overall variance between the individual data sets, and then looking at the meta-analysis data set in the middle gives you an idea that the meta-analysis is a good compromise between the variability of the PCA in the individual data sets. To further dive into some of the differentially expressed genes, the top 5 at the meta-analysis are shown on the top in the upregulated, and the top 5 in the downregulated are shown on the bottom. These genes are actually genes that are highly regulated in some of the individual data sets, so it's no surprise that these would come out on the top of the list. But what we show here is that we are providing not only the log 2-fold change, but also the 95% confidence interval, as well as the p-values for the meta-analysis and adjusted p-values as well. So this gives us an idea how much log 2-fold change there is, but also an insight into the confidence interval for these genes. And as I mentioned before, another way to visualize this would be a forest plot. So what we're able to do with this data set is look at individual genes such as LCN2, which is also known as NGAL. And on the y-axis, we show the individual data sets. And then on the x-axis is the log 2-fold change. So what you can do is look at the individual data sets and see their log 2-fold change in their variance. And then in the red, what we see is the results of the meta-analysis. So while there is some variability in the log 2-fold change, in this case, all of them are greater than 0, but there is some variability. But by using the random effects model, we're able to combine those variances and fold changes into a single value and understand the confidence in which this is differentially regulated. The top downregulated gene is aquaporin 8, and similar to what we saw with LCN2, in this case, these are all less than 0. You can see a wide range of variation in the log 2-fold changes, but we are able to effectively summarize this in the blue with the meta-analysis results. So while we're able to show you a couple different genes, and while this is interesting from a point of view of the genes that will be most confident in showing their regulation, what we are going to provide upon publication is a web tool which allows you to search for individual genes and see their corresponding forest plots. So shown here, I just picked a gene, heregulin, which is a gene I worked in a previous life. And you can see that for the case of heregulin, there's actually very little regulation. As you can see, the confidence intervals are very wide, and the scale on the bottom indicates that there's not much overall change in the log2 fold change value. And then the overall fold change summary indicates somewhat of a compromise between all the other values seen in the individual data sets. So with this tool, one could search for any gene out of the 27,858 and pull up those forest plots. And as I said, I think this will be useful to give you a simple answer to the question of whether your gene is regulated by giving a single value. Now, to take a kind of a broader view of the question of the meta-analysis, we looked at pathway enrichment for the reactome pathways. So we took the meta-analysis DEG as well as the individual experiment or data set DEG and performed enrichment analysis. And what I'm showing you is 6 of the immune-related reactome pathways that come up as being enriched in either the individual data sets or the meta-analysis. So you can see, for example, for chemokines bind receptors, chemokine receptors bind chemokines, there is enrichment in most of the data sets to a large extent. And in this case, for GSC 114527, there's not significant regulation or enrichment, but you can see in the final column that the meta-analysis shows enrichment as well. And so for the upregulated pathways, most of those were regulated in the individual and the meta-analysis, with the exception of peptide ligand binding receptors, which shows that only 6 of the 8 individual data sets show regulation, but by meta-analysis, it shows that it is regulated. As an example of the down-regulated pathways, so genes that are down-regulated, what pathways are enriched, you can see there's much more variation between the individual data sets. And it's interesting to see that in some cases, such as fatty acid metabolism, only 4 of the individual data sets show enrichment of those pathways. Whereas when we do the combined analysis of the meta-analysis down-regulated DEG, we do get enrichment. So this is not too surprising, as the extent of differentially expressed genes for the upregulated genes is much greater than the downregulated. So you do see more subtle effects, and in this case, it's very useful to look at the individual data sets and the meta-analysis to understand those behaviors. So in summary, we've used a random effects model to integrate 8 UC microarray data sets while retaining within and between data set variation to be visualized in the forest plots. LCN2 and aquaporin 8 are the most up or down regulated disease markers in UC and meta-analysis. Chemokines and ECM pathways are enriched in the upregulated meta-analysis DEG. Biological oxidations are enriched in the downregulated meta dataset DEG. And importantly, this study highlights the need for increased standardization and reporting of patient-level data. For example, sample and disease severity information. Because while we were able to get meta-analysis results, we do believe if we had more individual patient-level information, we would be able to tease apart some of the covariates that are important for this. But as it is, most of the time, minimal information was provided about these samples and these patients. So lastly, I'd like to acknowledge my coworkers at Elementive, Vibel Jurath, Ju-Ai Zhao, Lisa Shackleton, Brian Fagan, and Niels van de Castile, and then the members of the Elementive Translational Research Consortium that were all involved in this project: Mark Silverberg, Shadi Nayeri, Dermot McGovern, Susanna Salas, and Bram Verstock. Thank you.